In this video, we will be exploring genetic engineering, which is also known as biotechnology. The learning target is I can explain genetic engineering and selective breeding. Genetic engineering is the deliberate modification of the characteristics of an organism by manipulating its genetic material. Deliberate modification means the scientists have changed something on purpose, and the characteristic of the organisms are changed by manipulating or changing its genetic material. The earliest example of genetic engineering is selective breeding. Did you know that all of these dog breeds are actually the same species? Every one of these dogs is a descendant of wolves. Humans started choosing wolves with traits they admired, such as coat color, personality, size, behavior, and started breeding them together for those traits. When different groups of people with different needs are breeding dogs for all different characteristics, you end up with lots of different variations of dogs. Selective breeding is when you breed only those plants and animals with desirable traits, meaning you breed plants and animals with traits that you like. People have been using selective breeding for thousands of years with farm crops and domesticated animals. For example, this mama cow has a trait I like. She makes a lot of milk. This papa cow has a trait I also like. He was quick to mature. By breeding them together, meaning I have them mate, their offspring will have traits of both the parents. These calves have both a high milk yield and are quick to mature. As a farmer, this is beneficial to me because they're going to mature faster, which means I can sell their milk sooner. This is an example of species influencing each other's evolution. As humans chose what traits to breed in dogs, they gave back to us and changed our behaviors, and we in turn changed their traits and behaviors as well. This is called coevolution, organisms evolving together. Another great example is the evolution of corn. The earliest ancestor of corn is actually this tough grain that isn't very sweet. But people in the Yucatan Peninsula in Mexico realized that if they started crossing or breeding the plants with the sweeter and softer kernels, they could get a sweeter and softer plant. After crossing these sweeter and softer plants over many generations, we ended up with modern corn. This modern corn helped feed thousands of people, so people lived longer and had more productive lives. This is coevolution. We affected the corn, and the corn affected us, and we have evolved together. A more technologically advanced version of genetic engineering is gene splicing, which is also known as recombinant DNA. This is the ability to combine the DNA of one organism with the DNA of another organism. Recombinant DNA was first used in the 1970s with bacteria, so this is a relatively new technology. To make recombinant DNA, a scientist must first remove the DNA from a bacteria cell. Bacteria DNA is called a plasmid, and it is in a circle, unlike human DNA, which is in a double helix. The scientists will use restriction enzymes that cut a specific piece out of the bacteria DNA. Next, the scientists will isolate a gene that they want in human DNA and use a different restriction enzyme to cut out that specific gene. These pieces are combined together with a third enzyme and inserted back into the bacteria. Notice this is a brand new combination of bacteria and human gene, hence the name recombinant DNA. Bacteria are asexual reproducers, which means every bacteria born of this parent bacteria cell will have the same DNA, this new combination of DNA that I want. And the foreign gene that I inserted will be expressed in the bacteria. Some benefits of recombinant bacteria are scientists making bacteria that can produce human insulin or human growth hormone. We actually trick bacteria into making medicine for us. People who cannot make insulin on their own have diabetes, but they can inject insulin so that they can survive. And we make insulin using recombinant DNA from bacteria in a lab. We have even genetically engineered bacteria that eat oil spills by altering their DNA. Let's look at this close one more time. Let's say I want my protein product to be insulin, so diabetics can inject this and stay alive. I'm going to isolate the gene, healthy gene for making insulin in a human and cut it out using restriction enzymes. I'm going to cut and paste that into a bacteria plasmid with a chunk missing to make a recombinant DNA. I'll place that recombinant DNA back into the bacteria cell, and now this bacteria will go through transcription and translation to make human insulin that I can give to my patients so they're not as sick from diabetes. The DNA of plants and animals can also be altered. We have used recombinant DNA to make disease-resistant and insect-resistant crops. For example, this corn has had a gene for a poisonous beetle inserted into its DNA, so if bugs try to eat it, they die. This poison is not effective on humans, which means we get to eat the corn and the bugs do not. We have also made it so fruits are hardier, meaning they last longer in our refrigerators. This is so effective that 70 to 75% of food in our supermarket is considered genetically modified. For short, we call these GMOs, genetically modified organisms. Genetically modified animals are called transgenic. Some examples of transgenic animals include mice. We have inserted genes from unhealthy people into the DNA of mice so that we can study how the sickness affects their bodies and even test new medicines. We have genetically modified chickens to be more resistant to infections, so we can grow them faster and fewer die to disease before we can eat them. We have even genetically modified cows so that they make more milk and leaner hamburgers for us.